Good morning, everyone. Welcome back to our course Genomics and Proteomics. We will be dealing with lecture 3 today. That's Introduction to Transcriptomics and RNAomics. So the lecture I have divided into 3 sections where you can see here. So each block represents a concept that I will be discussing today. We will go back into a little bit more basics again. We will talk about gene expression, central dogma of gene expression. And we already know that there is a few exceptions to the classical central dogma theory. And then we will talk about a little bit more of types of RNA. It's not only mRNA, tRNA, or RNA. We have many more RNAs in our system in general, the cell biological aspects. We will talk about that. And then we come back into a definition or transcriptomics, the scope of the study. And then we will jump into a little bit more and the aspects of differential gene expression, alternative splicing, quantification of gene expression. At the end, we will talk about how we can correlate or study functional aspects of transcriptomics in terms of diseases. So again, I'm saying that the class is an introductory class. So I would like to introduce to the concepts. And again, more technical details, we will go up into the curriculum and then take each concept and then learn in more detail. So this is the classical central dogma, right? The view we know about gene expression. We start with DNA. DNA undergoes into DNA is transcribed into RNA, and RNA is translated into proteins. So each step we call it as transcription and the translation. So it was considered at the beginning as a classical view. At the beginning of the study, we know that it's a unidirectional, and then one gene, one enzyme, or one gene, one protein codes for specific gene expression studies. That's what the classical view of gene expression or central dogma. So coming on to the diagram here, you are looking into a diagrammatic representation of a cell that shows outer cell membrane and then inner nuclear membrane. It's a eukaryotic cell. And then it shows about the same concept here of the double-stranded DNA is converted and transcribed to become mRNA. That pre-mRNA will be here. You should remember that. mRNA undergoes a processing step. Pre-mRNA will be formed within the nucleus and then it becomes mature. After maturity, the mRNA will be transported into the cytoplasm and where it will be translated into proteins. It should be remembered that the DNA will be always residing in the nucleus, but the RNA kind of localization changes. And the change in localization is also an important aspect of its functional significance that we will discuss in further classes or further slides. We will see how we can do that. And this is classical view, right? We are talking about this concept. But the view has been changed. So the concept has been questioned. This is the hypothesis. People tested, people confirmed it, people were following it up a quite long time. But there is again some exceptions or evidences that you don't need to DNA to express a gene or protein, or you don't need an RNA to express a protein or gene. That you can eliminate a step above one step each. You can go out of DNA, you can go out of RNA, you can have only protein. Still, you can express a functional protein into that next generation. An example is prions. We will see in the next slide. One example of taking only proteins, the functional protein that is inherited into the progeny and it gives the functionality. That's prions we studied, and we know that that's one of the exception cases. And if you take up this step alone, take the RNA alone, leave the DNA. Yes, we know that many of the viruses, RNA viruses, they don't have DNA at all. They have only RNA, and then they can still go ahead making proteins, and they can produce the genes and proteins functional survival and its replication process. So we know that each step can go independent of 1, 2, 3. Then you can go 3 to the end function, go step 2 and go to the function. And in terms, one concept that recently discovered is DNA methylation, epigenetic marks on the DNA. Irrespective of whether the RNA and protein influencing the function, a methylated mark on the DNA can also influence a specific phenotype of the progeny. So that's been already discussed. So in simple terms, this slide, DNA is transcribed into RNA, RNA is transformed to mRNA or pre-mRNA, RNA, then mRNA is translated into proteins. So the exceptions, what I discussed in the slide, they are discussed here again in a representation. Each step cannot independently can regulate an expression or protein formation. That becomes exceptions to central dogma. And we already know that another process called RNA viruses, they can go ahead into a process called reverse transcription. This is transcription, and they found an enzyme reverse transcriptase that can move it back from RNA to DNA. So a process can be not only unidirectional, this step can go back in the bidirectional way. That also has been established. Coming on to types of RNA. It's not only one mRNA that exists in the cells, there are many RNAs. For example, you see a different table that represents from the Brooks and Cloday as Image Learning Center. So the RNA type is here, the size, because it's a nucleic acid, so ribonucleic acid. And it should be, we already know that it's single stranded. So we don't talk about base pairs in RNA when you talk about either bases or you talk about residues. So each function, then you can correlate here. For example, messenger RNA, that directs amino acid sequence of the proteins. That already we know that. mRNA codes, codons consist of that codes for specific amino acid, then they become proteins. But in addition to that, we already know transfer RNA, ribosomal RNA, but few other new RNA molecules that came across. For example, one example here, microRNA. This is also very important for gene expression, alternate to producing proteins. On its own, microRNAs can regulate a gene expression process that is important for growth and development, and also in diseases, and more information is coming along. So going back into classical, the structural or linear representation of the DNA and RNA, how the mRNA process, pre-mRNA processing happens, just an example, again, graphical representation. Look at the genome level, double-stranded DNA here, goes to RNA and the protein level. We already know that in RNAs, the process will happen with pre-mRNA. There are exons and introns, mRNA precursors. The splicing phenomena will come, and then introns are removed, only exons are combined, they go into in terms of making proteins. That we already know, we studied. And coming on to not only mRNA, other RNAs, these are called non-coding RNAs. NC means non-coding, that may be in the intronic sequences. So there are many types of non-coding RNAs. This is probably microRNA, and then tRNA, rRNA, and they will have a different looped structures that you can see it here. And they might have specific, they also have specific RNA modifications on the nucleic acid residues. That also gives a structural or important identity for the specific non-coding RNAs. So these can be called ribonucleomes, transcriptomes, and that gives rise to proteome. So initially we thought only expressing of mRNA is only giving protein that is important for regulatory function of any gene or genome, but it should be noted that intronic non-coding RNAs, it's not exceptions. What we discussed as prions or RNA viruses, a lot and a lot of functional studies are coming, and it's been confirming that non-coding RNAs possess very high regulatory challenges. They pose regulatory challenges. They control a lot of regulatory function of gene expression in our cells. So it should be noted that they are not exceptions at all. It's really important for our gene expression studies. So this slide represents a specific in terms of quantification, or in terms of how many genes are there in the system and how many genes are there in a human genome, and how many of them are coding for specific RNA molecules. We talked about the types of RNA in the previous slide. So let's say if you take a genome, in the genome, how many genes are there that will regulate or that will code for specific RNA molecules. This chart represents, this was around 2004, the article has been published. You can access the article here with the DOI number. So when they analyzed the human genome, after Human Genome Project completed, sequences are available, they analyzed it for specific molecules of RNAs, and they were able to see that in the genome, in the whole genome, protein coding genes are close to 20,000 to 330. That is, we already know that 20,000 proteins are there in our human genome that codes for that. They could see corresponding genes for that 20,000. But in addition, they could see several components of this other RNA molecules. For example, long non-coding RNA genes, small non-coding RNA genes, a kind of 13,000 here, kind of 9,000 here, and of them, there are different categories. For example, small non-coding RNA, one of them is called microRNAs, another one is other small ncRNA. And in terms of length or in terms of size of the RNAs, so the information has been known that why these are called small non-coding RNAs, because these microRNAs are very interestingly 25 nucleotides long only, not more than that. We are talking about residues, 25 nucleotides, in case 21 to 25 is the range. They are very small RNA molecules. And the long RNA molecule, non-coding RNA molecules, can they range from 1,000 to 10,000 bases, excuse me. And the same way, the protein coding, depending on the gene, the length and size of the mRNA changes. So it should be noted that there is a repertoire of RNA molecules in a cell, and the different sizes, different functional, regulatory and functional roles, and the proteome production roles are there. So here comes the study of transcriptomics. So transcriptomics is the study of transcriptome, the whole RNA molecules present in a cell. That's called the complete set of RNA transcripts that are produced by the genome under specific circumstances or in a specific cell using high throughput methods. That is the definition of transcriptomics. We are looking for a specific RNA transcript available in a cell, either that in a specific circumstance, in example, in a disease condition or in a developmental condition, larva, adult, or it's going through a molting insect, what is the molting, molt 1, molt 2. There is different larval stages that you can look into that. What are the RNA transcripts present during that stage or present during a disease condition, that is either cancer or either other lung disease, something else you can take into an example. And it's also omics study, so it uses high throughput methods to analyze the data. Okay, we introduced two different concepts of types of RNA, and then we finished with what is the transcriptomics, what is the branch of biology that studies. Then you go into a little bit more functionality of transcriptomics. You need to understand how RNAs regulate the development or how RNAs regulate the gene expression in a given specific cell. So one of the concept we already know that differential gene expression or tissue-specific gene expression is the hallmark for the development of a highly organized animal like human beings, different tissues, different organs. We may start from a single cell that is having the same complement of the genome, but as the cells go along, we get a different fate and different RNA transcripts that move along. Along with that, we will talk a little bit about alternative splicing briefly, and then methods to quantify RNA transcripts. We'll go ahead. So, for example, you take you just think of how many cells are in a human body. How many cells are there in human body? A question you can pose yourself. And then how many mRNAs are present per cell? And also you can say, will the mRNAs produced in cell 1 differ from cell 2? You can ask these kind of questions. This gives to fundamental foundation for differential gene expression. Yes, the answer is how many cells are in human body? We are made up of trillions and trillions of cells. Cells which have a different function, and cells based on the tissue level, they have a different functionality. For example, in terms of also tissues, in terms of morphology, we have a huge diversity. In terms of immunology, we have lymphocytes, T lymphocytes, B lymphocytes, macrophages, and other systems, other cells that help in immune system. And also, in terms of, for example, nervous system, the morphology of the neuron is quite different, right? The neuronal cell has a very diverse morphology. Trillions of cells, and on average, a cell can produce approximately 10,000 mRNAs will be there in a cell. That is already established. People know that. And yes, if you take a scenario of cell 1 and cell 2, a set of mRNAs or RNA transcripts or transcriptomics may differ from cell to cell. It may differ from population to population. It may differ from wild type situation to a disease situation, or wild type scenario to mutant scenario. Yes, that will be already, we understood that process. So I'm giving you an example of a developmental event during Drosophila, the fruit fly. They are trying to understand the gene expression studies during embryo development of the Drosophila. So this is a Drosophila embryo, and you are looking here earlier development, later development. You are looking into anterior, posterior in terms of space, in terms of what they observed that when they try to understand a specific set of expression profile or an example specific set of the genes, there are specific set of genes in Drosophila that control anterior posterior patterning. They're called nanos, bicoid, giant, cripple, even skip, and wingless. So they were interested to study how these genes are regulating this anterior posterior patterning. So they try to understand where the genes are located in the embryo. They worked on a technique called in situ hybridization or mRNA localization. They raised mRNAs for nanos through the gene and genome that gives a specific mRNA. They were trying to localize those mRNAs or profiles into the embryos. They could able to see that these genes during the developmental time have a varied expression along the anterior posterior axis of the embryo. So that gave them an important aspect that, for example, bicoid is here in blue, nanos is here in the posterior, bicoid in the anterior. This specific localization of the genes or specific production of mRNAs in a specific location from the genome is important for pattern formation. That one aspect they understood. And then as more development progresses, there will be more segments will form. In each segment, there is an even and odd segment. Even skipped present is only in the even skipped or even segments. Then there is a wingless that comes along. Then wings, legs, and other thorax phenomena that parts of the body will form from the embryo. And the pattern has been established from the beginning itself. So this one way to study gene expression, study RNA transcript. In another way, one more study they studied on the zebrafish development of circulatory system or blood circulatory system. And they looked at two scenarios. They had one wild type scenario here, two wild type animals. Then they have a specific condition called a notch mutants. The notch is a specific important protein that regulates several developmental events all over the animal kingdom. So they looked on a wild type situation where notch is normal, wild type functioning. They looked for a presence of arteries and veins, where is the notch localization is there or notch is expressed or not. They used a method called in situ hybridization, and they localized that notch is present in a wild type scenario, both in arteries and both in veins. So they observed that. And when they observed a scenario that when what happens in a situation there are notch mutation happens in the genome and the notch proteins are not expressed, then they are called as notch mutants here in this posterior, in this right side of the slide. They observed that the expression of the transcript of the notch has disappeared from the arteries in the animal. And simultaneously, very interestingly, they could see that veins were produced double the amount, double of the veins are there. So probably there is a functionality of the notch that it regulates a proper number of the veins and arteries to be produced during development. If a mutation happens, arteries are, it's absolutely necessary for artery development and to control the number of veins. Okay? So they use an in situ hybridization. Excuse me. So coming back to next system, what we talked about. So developmental profile, we looked at two examples, Drosophila development and then circulatory system development in zebrafish. Then one more an important concept is alternative splicing. That's very important. We might have discussed in a few classes before. There is a splicing phenomena that happens that takes the introns and then only exons are combined that gives rise to specific proteins. And we know that there is another mechanism of splicing where some of the exons can be skipped or they may be left, and the splicing goes in an alternative way, exon 1 and exon 3, or exon 2 and exon 4. There may be elimination of the exons. That gives rise to alternative splicing, mRNA variants, that gives rise to protein isoforms. That has been known already. In a case, exon 1,
Exon 3 and exon 4 are the confused.
Yes, yes, we will see that here. So all exons are there, 1, 2, 3, 4, 5, a specific protein isoform A can be formed. But when exon 3 is skipped, there is an alternative form of the protein. You see that one of the helix or one of the structural part is missing. Then you get into a protein B that is missing this portion that is coming from probably exon 3. Then you keep the exon 3, you skip exon 4. Only this helix portion in the anterior will remain. The other helix portion can be removed. So this is the protein isoform. Same mRNA can give different splice variants that gives us to different functional or isoforms that gives us to different function. And it should be noted that human genome encodes a lot of alternative splicing. We are spliced through alternative splicing, and that gives rise to different isoforms. One of the transcriptional regulation in humans through alternative splicing. So coming on to the methods. So in this present class, I may not go too deep into the methods. We will talk about in another class where each technique comes up. But to give an overview or glance of the situation, how the RNA can be detected, there are many techniques we can use for detection of RNA in a level of single RNAs, single gene. You go into a cellular level, you go into a whole organism level and population level. You can go ahead. In situ hybridization, Northern blotting, PCR, RT-PCR, qRT-PCR, RNA-seq, and microarray. These are the techniques that are used for analysis of the RNAs. With a sample size, you can go ahead with from one cell, one RNA to a microarrays. A population of genome transcriptomics can be studied here. I will briefly go up with the mechanism, how it works. So let's see in this diagram, we have a transcription happening in the nucleus, cytoplasm. The genome has been unwound, and then there is antisense, sense strand, RNA polymerase goes up, it makes the RNA. We already know that the DNA replication happens, and there is hybridization with that. There is a ATGC pairing, and we already know that DNA RNA hybrid also exists in the cells, and the hybrid goes with where A will bind to uracil RNA, not the T in the DNA. We know this specificity of the hybridization. And also this can be used for a mechanism of probing a specific mRNA molecule that we will see in the next slide. And also one more important thing, if you don't have DNA, you can move from DNA to RNA. But if you don't have DNA, you start with RNA, you can make the DNA copies of the RNA. That is called complementary DNA, where RNA is converted to DNA. In a system RNA viruses, they exist only RNA. You can use reverse transcriptase through RNA-dependent RNA polymerase. You can make complementary DNA. This DNA and RNA hybrid, and also it will help in specific processes of quantification in RT-PCR and qRT-PCR studies. We will look into that. And also, if you are making a probe of DNA, for example, you make a probe of DNA and you want to identify a corresponding RNA. If you don't know also, you can take a stretch of the gene, make the nucleotides with the fluorophores. These are the things, lightning or the green or red fluorophores attached to the DNA probe. They can go and detect this. When the DNA RNA hybrid forms only, the detection gives a glow and the signal will come from the fluorophore. The hybrid should be required to give a signal. So based on that, based on the quantification, how many probes are there, how many lighting signals are coming, so you can quantify how many mRNAs are present in a system or a cell. So again, agar-gel electrophores is used like DNA. So this is an example of, these are the molecular weight of the markers here, 9 kilobases, 500 bases. Then you see an intact RNA isolated from the whole cell and degraded RNA. It should be noted that when you go ahead with isolating RNA and DNA from a cell, DNA is a double stranded. It's probably supercoiled in many ways, and it is protected of degradation because of the double stranded structure. But RNA is mainly single stranded, and then will be degraded easily. There is RNases that can degrade RNA. And you can see that if you don't isolate the RNA through proper position, it is a degradation of the sample happens. Then you may not get a good quantity of the RNA of the whole cell or system you are working. Then you may unable to detect the sizes of it. But if you do a good process of isolation of RNA, then intact RNA can be found, and through the agar-gel, it can go through different processes of based on the size. 28 as 80. For example, if you take an example of microRNA that maybe already has passed out of the gel because of the small size, there are different methods that you can go ahead. So we have close to 4 minutes now. I will quickly go to 2 slides where one more study of microarrays are really important. The microarrays are, you're not looking at a single RNA isolated from a cell or a single gene or protein. You're looking at the whole organism level, cellular organism level, that you look into that. A microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. The same time you can detect. You don't need to run a single gel for it. You can detect whole set of genes at the same time. DNA microarrays are microscope slides, the slides that will have printed with thousands of spots of DNAs here. You see that it's a chip. Different spots of DNA are just spotted on that chip as the DNA probes, and then each DNA codes for a gene. And then these gene chips or DNA chips are used to probing specific transcript levels or attaching to the specific complementary mRNAs present in the system. So I won't go much detail into the how microarray detection system works. Just giving you a glance, this is the DNA chip. These are the DNA probes that will catch to specific transcript level within a cell or a system. Then at the end of the detection, you see a kind of red, brown, black signal spots for each spot. Then we will see why the green signal comes, why the brown, why yellow, and why other concepts that will have a specific indication of the expression level or availability of specific mRNA in the cell. That we will discuss in further classes when we take up microarray and RNA-seq for one class only that we will study. So comparison of transcriptomes allows the identification of genes that are differentially expressed in distinct cell population or in a response to different treatment. So you can, using the transcriptomes, you can compare the system, see how genes are differentially expressed. For example, you're looking in a cell population or you're looking in a disease condition, you can compare the transcriptomes in wild type condition or without disease, compare with disease and see and make a conclusions out of it and find a remedy or find a drug that's suitable to that. For example, you take in a case of microarray analysis done through for a disease of breast cancer cases. So, and also you should remember that we spoke in omics studies, lot of time there is quantitation, statistical methods, bioinformatic tools will come up. So you need to be quite good at statistical analysis also, because the data will be huge. So you will have a huge data about it. For example, this is a clustered correlation matrix of 266 breast cancer cases. So you can see a chart that gives a specific color indication, upregulation or downregulation of the genes. You look at a specific case of gene GATA factor here, T10 here, and other genes. In a specific population of 266 cases here, you go by lineage. In which person the case of scenario is like this, in which person the case of scenario where upregulated, where are the mutation. Which tissues, example, lungs, breast cancer usually go into look into specific tissues and upregulation, downregulation of the gene. That's one of the way you can study into that. Okay, so we briefly stop now and we will stop the lecture. So go back to the definition of transcriptomics here. And then we will take up tomorrow proteomics and metabolomics 11, 11, 20. But try to understand the general aspects of transcriptomics for today's class. Okay, thank you. I'll see you tomorrow, 11:20.